Hello, welcome to the screencast where we'll be discussing Drosophila melanogaster, some background and a little bit about the life cycle and why they make excellent organisms of choice for studies in genetics. So by the end of this, hopefully you'll be able to describe and explain the life cycle of Drosophila melanogaster, why D. melanogaster makes a good model organism for genetic studies, and then just a little bit about the genome of D. melanogaster. Okay. So here is Drosophila melanogaster. Undergoes a complete metamorphosis life cycle, and it's around 2 weeks. So these are good organisms of choice because, for many reasons, to do experiments with. One is that they are relatively easy to care for. Right? They require very little in terms of maintenance. You can, pardon me. You can house a lot of them in a small area, and here's the culture vials over here, and we have some of those in class that you've seen. Right? There is typically low ethical, legal, or social implications. LC here, ethical, legal, social implications. They are invertebrates, so there's not a lot of protests about using them in studies. We can dispose of them as we need to. We can keep many of them. As I said, they are inexpensive, right, to keep, which is a consideration if you're doing studies here. So let's talk a little bit about the life cycle, and then get into some other reasons about why they make good organisms, specifically for genetic studies or studies of heredity. They are. You can see here's the female, right, and here is the male. So the females are larger than the males, and this is, they are sexually dimorphic. So you can, with some practice here, you can relatively easily tell the males from the females. Females are larger. You notice that there, and we'll have a slide specifically about telling the difference between males and females. But this means that you can basically control the matings. And so if you are you control the matings or control the crossing, so you know which two individuals you're crossing with whom, so you're able to track then through many generations the trait. So speaking of generations, you can get many generations in a short period of time. And again, like I said, their life cycle is about 2 weeks. They produce many offspring with each mating, and so this gets you a large sample size, right, to then be able to run some statistical analysis, right, to further support your hypotheses. Again, just let me go back to this mini generation thing here a second. So many generations mean this is good because then you can follow a trait and how it's passed on from one generation to the next. As I said, they're sexually dimorphic. You have lots of offspring there. And let me just pause here now and just kind of talk about that life cycle a little bit. So when the adults here, and the biological meaning of adult is that they are able to reproduce sexually. So the adults, as they emerge from the pupa, are very soon ready to mate, and the male will court the female, and you can actually watch this courting behavior happen in the vials if you're patient enough. More on that later. So the males mate with the females. The females will then lay eggs down here in the medium. Right? So this bluish stuff down here is the growth medium, and there's a fungus that kind of grows on that medium, and the adults will, the larvae then will feed on that. So the female will lay her eggs in the medium. Those eggs will then hatch into an embryo. Those embryos then will undergo these changes into the larval stages, right? So this first instar larva, the second instar larva, the third instar larva, right? And in the vial here, you can see the larvae. So the larvae are these lightish, creamy colored things here, and you can also watch the vials, and you can see the larvae sort of crawling down all through the media. And so these larvae are basically eating machines here, right? And so if this was a butterfly, the larva is analogous here to the to the caterpillar, right? Then the larvae the third instar larva will begin to then come out of the media and crawl up the sides of the vial there, and you get these prepupa, which are lighter, and then the pupa, which are darker. So you see the pupae here. There's two of them, and those pupae then stop moving. They get darker and darker and darker, and then out of the pupal case will emerge the adult fly, either the female or the male, right? So this is basically the life cycle here, a complete metamorphosis going from this larvae into an adult, and the adult does not look like the larva, right? That's the complete metamorphosis, right? So they are, again, we said that they are ideal organisms for genetic research. So again, they're sexually dimorphic, and they are diploid. So here we see the male copulating with the female. You notice that the difference in the males, so the abdomen here, you can tell the abdomen is much darker in the males. It's a little bit pointier as well. The female abdomens are a little bit more rounded and not as dark, right? So again, females and males, you easily isolate a female and a male that you want to mate with one another, so you can control the matings. This makes them good for genetic studies. Another way to tell females from males is by looking at the forelimbs here. So on the females, these structures are absent, but on the males, the structures here are referred to as sex combs. So this is you're able to tell the sex or the gender, right? Because the male has these little sex combs on the forelegs there. So a couple different ways to tell the difference between the males, and the females. Another good reason that they are excellent organisms of choice, and this I think is number 7, is that they have many easily observable phenotypes. And so we're comparing them to the wild type fly here, and the wild type fly you can read about on the Fly Lab website, but the wild type fly will have red eyes, has a darker body here, right? The wing shape is wild type, and again, wild type is what is considered your normal, right, or basic fly, right? So some examples of mutants then would be ebony body color, right? So this is a mutant, right? And we abbreviate this, the mutant here as EB, right? And the wild type is just a plus. So your wild type for body color, you'd be plus, and then the mutant would be ebony. Another body color mutant is yellow, right? And you just use this Y as the notation there. Here's another easily visible phenotype, and these are vestigial wings. And so you notice that the wings here are sort of all shriveled up. These are especially good flies to work with because they don't fly away on you. These other guys will fly away, so you have to anesthetize them. But we can practice with that if you're willing to help sort of subculture them. And again, just the insignia there, vestigial is VG. Okay, so lots of easily visible phenotypes. These are just a few samples of them, few examples. Another thing that makes Drosophila melanogaster relatively easy to work with is that for genetic studies is that there are only 4 chromosomes, right? And so the genome are relatively small, relatively easy to manage. And so you see the first set of chromosomes are the sex chromosomes. So sometimes these are indicated as chromosome number 1. But you have an X and then a Y. In this case, the Y is longer than the X. And this is reverse as it is in humans, right? The Y chromosome is much shorter than the X chromosome. And then chromosomes 2, 3, and 4 are the autosomes here. And you notice that 4 is this real stubby little short guy, right? And so millions of base pairs here is the unit. Right? You also have this unit here. These are centimorgans, right? And more on that in another screencast. But you see centromere positions here. Right? Here's just a graphic sort of the chromosome. So chromosome X here. So this would be a female, right? Inherited 2 X's, right? And then chromosome 2, right? 3, 4, and again, you notice that they're diploid, so this is a homologous pair here, right? And then this would be the karyotype from a male over here, right? Because you have the X and the Y. And again, the Y is going to be longer here, and so it's kind of bent, curved over right there. Right? So a little bit more about the genome here. The genome, another thing that makes them good organisms of choice is that the genome has been sequenced. So we know the sequence from the tip of this X chromosome all the way to the end of chromosome number 4 has been sequenced, and many genes have been mapped to those chromosomes. And so this is what you see here is a map of where the different genes are, right? So for instance, the star-eyed gene is on chromosome number 2 at this locus right here, right, etc. Right? Curled wings on chromosome number 3, it's at this locus, right? And these give you relative distances from the tip of the chromosome, okay? So we know an awful lot about this organism from having studied it all the way back to Thomas Hunt Morgan, starting in Columbia University in the early part of the 1900s, right, 1920s, 1930s. And lastly here, we mentioned that the genome has been sequenced, and there's a whole website here maintained by National Human Genome Research Institute, which is part of the NIH, right? And this one happens to be from Indiana University. And so you can search this database, right, a database of Drosophila genes and the genomes, right? And find out a lot of information about the genome of the fly. You can do comparisons, etc. Right? So the fruit fly has given us a lot of information about genetics, a lot of information about chromosome structure, about gene regulation, right? And we owe a lot to that little organism there. And we have some in the classroom, and I encourage you to take advantage of maybe a broom block, or coming in and working with them, getting used to handling them, maybe even doing some breeding experiments of your own. Okay? So hopefully now you're able to here describe the life cycle of Drosophila melanogaster, why Drosophila melanogaster makes a good model organism for genetic studies, and we had sort of at least 9 reasons, right, there, and maybe you can come up with a couple more. And then just a little bit about that genome, the structure, basics of the fruit fly. Okay, thanks a lot for listening. Bring your questions to class.